So our next speaker is Marcos Garcia Fuentes from Department of Pharmacology, Pharmacy and Pharmaceutical Technology, Universidad de Santiago. And we're talking about hydrogels activated with microRNAs for priming cell differentiation.
Well, thank you very much. Thank you very much for staying until the last talk of this session. And I'm going to talk a little bit about hydrogels activated with messenger RNA for priming cell differentiation. And well, I'm going to touch a topic that it was already addressed in the first presentation today, and the big question that we need to study is: do cell therapies comply with the requirements for medicines, or are they really safe, and are they effective? And if you go, of course, there are many therapies that they are approved, or a few therapies that they are approved, and they are effective with no question by clinical standards. But if you go to most of the studies that have been made, you can find many studies like this. This is a meta-analysis of many studies where cell therapies have been used, in this case for refractory angina. This is just an example. I could have picked up many other studies. And if you go to the parameters of response that they were analyzed, you can see in the studies that the responses are typically very much like in the border of being significant, maybe not very significant. And this goes to this very recent publication that the FDA, or some scientists from the FDA, drafted in the New England Journal of Medicine. And I would just like to highlight this sentence. Outside a few well-established indications, the assertion that stem cells are intrinsically able to sense the environment into which they are introduced and address whatever functions require replacement or repair is not based on scientific evidence. So it is very clear that we can just not use stem cells by themselves to treat most of the things that we need to address. And we need to provide these cells with the cues to really sense or react to this environment that we need to target and to treat. And this is very clear, for instance, in cases like cartilage, because cartilage, if you know, this is a tissue where there are no really very good replacements, and if you want to repair cartilage, there's not really an ideal material to do that. And this means that we really need to educate and to prime cells to differentiate into something that can repair cartilage. So if you try to think about what kind of things can help you to educate a cell to become a cartilage, a chondrocyte, you can see that there are a lot of pathways that you can touch. But one of the important things of many of these pathways is that they converge over a transcription factor that controls most of the chondrogenic differentiation processes. So one of our ideas was maybe we should just prime this chondrogenic differentiation pathway by introducing this transcription factor. Well, the problem with this is that these transcription factors, they are intracellular proteins, and you need to deliver them there. How can you do that? Well, you can use plasmid, you can use viral vectors. Ideally, you want something that is biocompatible and safe. Sometimes viral vectors are not ideal for that. You need something that provides high percentage of transfected cells, which sometimes you cannot achieve very well with plasmids. And it is important to have a time on-off protein expression, because maybe you don't want to enforce this transcription factor expression for too long because maybe you are blocking some maturation pathways. And because of that, we thought that messenger RNA is an ideal candidate to really prime this process. And we were unfortunately not the first to think about that, because this paper came in 2010 and they performed this lineage priming using messenger RNA, and this was in 2010 selected by Science as one of the top 10 breakthroughs. But we thought that, okay, if these guys were smart enough to do this in 2D, we could do the same thing, but in a 3D scaffold. And here we have our idea of a hydrogel or a matrix that is activated to perform this priming or differentiation process in situ by providing these transcription factors in the hydrogel. We call that the MR matrix. This is just a name for that we gave to this kind of technology, and it's very simple. It's just an hydrogel, and we will have some complex with a transfecting agent and the mRNA that should code for the transcription factor that we want to introduce. The materials, we have used several polymers. Most of the things that I'm going to show you are based on fibrin. We have used also collagens and other kind of materials, and you can see that you can introduce the complexes of the mRNA and they distribute homogeneously. So one of the things that we wanted to test first when we wanted to investigate this kind of technologies was whether the MR matrix could force the transcription factor expression. So, okay, this was the idea, but we needed to test whether this was true or not. And for that, we used mesenchymal stem cells from humans, and we transfected them with lipoplexes, either with mRNA or with plasmid RNA as a control, and with 2 transcription factors, SOX9 and MIOD, and we have also 2 concentrations of fibrin. And the most important thing that you can see here is that you can achieve very high overexpression of these transcription factors. See here, you have like 200,000-fold increase of SOX9, much larger if you use messenger RNA if you compare to plasmid DNA. And if you go to MIOD, which was another transcription factor, in this case we use a chemically modified messenger RNA, you can see that you can go even over 1,000,000-fold increase in a transcription factor. So with this, well, we also wanted to see a little bit of the kinetics. The messenger RNA expression decays slowly, more slowly if it is not chemically modified. If it is chemically modified, it can last for even, I would say, several days to weeks even. If it is not, it will decay over maybe the first or second week. The plasmid is more stable. The second thing that we wanted to address once we knew already about the expression is whether this imbalance in transcription factors could promote a change in the differentiation. And wanted to test that in the chondrogenesis. So we tested an MR matrix with a lipoplex with messenger RNA coding for SOX9 against another lipoplex with a plasmid DNA coding for SOX9, or a non-activated scaffolds, and again, well, the rest of the conditions were more or less the same one. And you can see here that you can achieve very high, this is after 21 days with no other chondrogenic factors, you can see that there is an effect on SOX9 on both RNA and DNA, even after 21 days, that the aggrecan was, well, high. Collagen type 2 was only significantly increased for the case of RNA. And interestingly, we also observed that there was, well, more or less, similar levels of collagen type 10, which is a marker of hypertrophy. So this indicated us that there was some chondrogenic effect. Here you can see some histology, again after 21 days. Here you can see that there was a very clear blue staining for RNA and DNA, which marks the presence of glycosaminoglycans, and also pink staining that is typical for this serious red, which marks collagen. So the next thing that I wanted to test whether these matrices could benefit or support chondrogenic medium. So if you add a chondrogenic stimulus like TGF-beta3, whether the presence of this messenger RNA could change something. And again, well, here you have the groups with messenger RNA, plasmid DNA, non-activated scaffolds, and we also added a pellet culture, which is the standard way to generate cartilage according to the science paper. Well, there's a gold standard method of generating cartilage, let's say. So here you have the results. Again, gene-activated matrices produce much more of the transcription factor. And you can see that all of the conditions, either with RNA or DNA or the pellet, generate aggrecan. RNA is, again, the best to produce collagen type 2. And one of the interesting things was that both of the gene-activated scaffolds produce much less collagen type 10 than the pellet culture. And as I told you, collagen type 10 is a collagen that you want to avoid because it's a marker of cartilage hypertrophy, of very fibrous cartilage that is not good for repairing joints. So this was an interesting result for us. And again, here you can see marked with Alcian blue the presence of glycosaminoglycans in the pellets and the DNA. And here also, this is immune staining against collagen type 2, the mark of presence, particularly in the RNA samples of collagen type 2. So another thing that we wanted to test whether we could use the same kind of matrices to prime other kind of differentiation pathways, in this case myogenesis. Again, we observe very high upregulations of MyoD, more than 1 million-fold. But look that there is also very high upregulations of MyoG, which is an early myogenic pathway marker, and a little bit in the myosins, which is a more mature myogenic marker. So, well, I'm not going to go over this. This is more or less, again, the same experiment, but with other type of myogenic medium. And I just want to show you some of the staining with for myosin heavy chain, showing that you can really generate cartilage myogenic samples with this kind of technology. And we are in the point where we are testing whether this kind of technology can be applied in vivo. We have just a pilot experiment where we have been able to support that this kind of technology is safe. We have no incidents or side effects in the rats where we tested it, and we have seen that there is muscle repair, but we are just trying to analyze whether this technology can provide benefits as compared to other competitive technologies. So we are not just there. So as conclusions, aromatics can induce remarkable transcription factor expression. We can trigger cell differentiation. We can support other differentiation stimuli, and we know that they are also safe, or they can be made to be safe under in vivo conditions. Thank you very much. Oh, yeah.
Thank you so much. The floor is open for questions. Any question? I have a question. It was not pretty clear to me. You conduct all these studies in static conditions?
Yes, yes, okay. Correctly, yeah. So
would you try to use a culture media in a epoxy media or epoxy culture? Have you tried to do as well to see the effect?
Yeah, no, we have not tried that, but of course there are many other parameters that we could try to understand how they interact with this gene regulation. So we have observed, for instance, this presence of TGF beta 3, or I didn't go into detail, but for the myogenic differentiation, we use some, we saw that the presence of some proteins also they can increase the myogenic expression. So of course there are many things in the culture conditions that can also affect.
Especially hypoxia.
Hypoxia.
Yeah. So maybe you could pursue that in the future. But very nice work.
Thank you.
So any other question? If not, let's conclude our session and thank you again, our speaker. I also would like to thank you all for attending and stay until the end. So thank you so much.